Okay, guys, let's turn our attention to some viruses here. This is all chapter 6 kind of stuff, and in the reading, I instructed you guys to look at a couple of those sections on viruses. So far, we've been focusing on the prokaryotes, broken down into archaea and bacteria. We compared those to eukaryotic cells. Now let's take a look at these viruses. Viruses really do strain the idea of what a living thing is. We have been talking about the smallest unit of life, which is the cell, right? But here we have an organism, well, not an organism, but a viral particle that is not technically considered alive. And that that definition, that being, you have to have cells to be alive, these viruses don't fit that bill, okay? So here we have some basic characteristics of viruses. It's a chart you can find in chapter 6 there. So it kind of brings out some of the main points about viruses. They are acellular, right? So they're not technically considered alive. And as such, as an acellular particle, these particles need a living cell to actually replicate. They need to hijack a living cell, whether it be a prokaryotic cell or a eukaryotic cell, and they specialize for the cells that they infect. They use the machinery of those cells to get themselves replicated. So they are obligate intracellular parasites. They are not going to infect a body or an individual bacterial cell and stay on the outside. They need to get into the cell, and they need to hijack those intracellular mechanisms. You can have DNA or RNA in viruses. It all depends on which virus you're talking about. But as far as structure goes, we definitely need to know the basic structure of a virus, just like we have with prokaryotic cells, okay? So here we have a few basics about viral structure. What we'd find associated with a virus would be a nucleocapsid, okay? This would consist of a capsid, which is typically proteinaceous material, so made out of protein a lot of times, and contained within that would be the nucleo part, right? The DNA or RNA. So here we have nucleocapsid, right, with certain proteins that are kind of arranged in a certain hexagon shape here. Sometimes these proteins are more in a helical shape like you see below, all right? So they are either in an icosahedral or helical fashion, right? Now, sometimes, though, you'll find that in addition to this, you may have an envelope, basically almost like a mini little plasma membrane, but it's technically still not considered a cell. We call it an enveloped virus, okay? So here we see a double phospholipid membrane, like you'd expect around any cell, right? But here we have this membrane with certain proteins in green here around this particular virus. This is an enveloped virus in this case. Down below you see another enveloped virus, and then within you have either your icosahedral or helical shape within there, okay? Now this is actually pretty critical information with the recent goings-on with coronavirus. Enveloped viruses like this do not fare very well outside of the body. This is a disadvantage for the virus, right? Because they want to survive, they want to replicate. So if there is an envelope associated with a virus, it's pretty easy to kill it. It's pretty easy to get rid of them and denature the proteins and break that virus up, because you can just apply soap and water or alcohols, things like that are going to kill the virus, all right? And this is exactly what we're doing with the coronavirus. The coronavirus is an enveloped virus, okay? Other non-enveloped viruses, they survive better on surfaces. Okay, they survive hand sanitizer and soap. Okay, but this coronavirus doesn't. So this is a good thing, right? And the problem is that enveloped viruses, once they are inside the body, they have a dangerous edge on non-enveloped viruses. They evade the immune system a little better than non-enveloped viruses. Non-enveloped viruses, the immune system can handle a little easier and get rid of them. And it's harder to make vaccines against an enveloped virus. Bad news for us, right? Because we're dealing with coronavirus right now. So this is really hitting home. This enveloped versus non-enveloped is absolutely part of the story with us dealing with the coronavirus. I think it was a couple months ago, everybody was super careful about what you touched, handles and things like that, and boxes that come from Amazon or whatever. But now we're finding that that's not quite as much a threat. I'd still be careful, but it's not quite as much a threat. But absolutely, person-to-person contact is something that you need to be careful of. All right, here we have an image. I just want to remind you that prokaryotes are cells. They also have a whole host of viruses that infect those cells. Okay, we usually call them bacteriophage. So bacteriophage are simply viruses that infect bacterial cells. Okay, so they also do exist. Now, how is it that a virus might interact then with a prokaryotic host? So here we have a prokaryotic cell. A virus needs to attach to that cell. Okay, so I want you to pay attention to these steps 1 through 5. I like to hone in on these kinds of things in exams, you'll find. I like people to know the steps in order of things like this. Okay, so we need the attachment of the bacteriophage to the prokaryote. Then there has to be a way to penetrate that cell. There has to be a way for the phage DNA to get inside of that cell, in other words. Biosynthesis would involve the production of the replication of DNA that are associated with that phage, with that bacterial virus, basically, and any proteins that would need to be produced to make new capsids, right? These proteins are going to go into the capsid part of the nucleocapsid for this bacteriophage. Step 4 would be the assembly of these viruses. So these viruses mature into actual bacteriophage. And then lysis, which would be the spilling out of these viruses out of the prokaryotic cell, most of the times killing that prokaryote in the process, and now you have millions of free bacteriophage to go on and infect new prokaryotes. Okay, so this is the release step, and I really want you to have those steps down, and you're just going to apply these basic ideas when we go to mammalian cells. When we go to eukaryotes, you're going to see pretty much the same thing going on with just a couple of slight changes in the words they use. Now, there is one other aspect that can occur besides the lytic cycle, where it literally lyses and kills the cell in short order. We also have lysogenic cycles. Now, there's some similarities, definitely. Here we have this attachment going on, right? So the attachment occurs over here, as you would expect. That has to happen. We have the penetration of that genetic material. So in blue here, we'll switch to blue. In blue here, we see this piece of genetic material has entered. But in this particular case, that piece of genetic material incorporates itself into the host genome that the bacteria owns. Okay, so here we have that circular piece of genetic material inside the bacterium, and the blue incorporated piece is actually viral DNA. Now, what can happen is there can be a period of time where these prokaryotes continue to replicate on their own, right? They're going through this binary fission that we're going to talk about more later. Basically, it's just the replication of all DNA and all contents of the cell to make 2 and to make 4 and 8 and so on. So this is effectively replicating the DNA of the phage for the DNA, for the virus, right? We call this prophage DNA, all right? Typically, a stress on the bacterial culture or on that prokaryote can trigger that incorporated piece to come back out of the circular genome and go into this biosynthesis, the other steps we talked about, the biosynthesis, the maturation, which is assembly, and the lysis of the cell. So all of those same 5 steps occur, except there's this gap and a prophage concept coming into play here. All right, another thing we need to really have a grasp on here when it comes to viruses, because they're so dependent on attaching and getting their genetic material into cells, they need to be able to attach well, right? So what we'll find is that viruses have, like here in green we see certain proteins happen to be on this particular virus. It looks to be an enveloped virus. And these proteins are specific for certain proteins that they have here in kind of pink here, I guess, right? That have a certain shape. Those green molecules are specifically going to bind to these pink Y-shaped proteins, okay? Now, if those Y-shaped proteins, like it says here, which are called the host cell membrane proteins in this case, if those proteins are only found in, let's say, chickens, well, this virus is only going to infect chickens, right? That particular species of chicken. If that Y-shaped protein is not found, then it will not infect. So let's say down here in section B, I don't see down here these Y-shaped red proteins. I see this orange-shaped field goal kind of shape, right? So these are not going to fit. This could be maybe a human cell, and that virus will not be effective against the human cell. It won't infect humans. So this is something that we would refer to as host specificity. Okay? Now, can that change? Absolutely. In fact, this happens all the time. The 1918 flu was caused by H1N1. Now those actual letters there do refer to proteins on the flu virus, and that virus was initially infecting birds. Okay, it was a bird flu. And then something happened, some change occurred to the proteins on the surface of that virus, and now is able to infect humans. So yes, host specificity can change. In addition to host specificity, we could talk about something called tissue specificity, or tissue tropism is the term they use. Again, let's go and talk about that coronavirus a little bit. The coronavirus, as we know it today, infects and causes respiratory disease, right? But so didn't one of its cousins, the Sars virus. The original Sars virus, a number of years ago, had a little epidemic of its own. It also had the same protein on the virus that allowed it to infect the respiratory system as the coronavirus does. Okay, so they're cousins. They have the same docking protein. They have the same protein for attachment, that first step that a virus has to make. However, the second step of penetration and getting that DNA into the cell was only found in the lung tissue when it comes to that earlier version of SARS. okay? With COVID-19, the penetration step is allowed to occur in the lungs, but the proteins that allow for the penetration step of coronavirus happen to be very abundant on all cells, and in particular endothelial cells, cells inside vessels, blood vessels, okay? So what we're seeing nowadays is not just pneumonia and some pretty nasty effects there in the respiratory tract, but in young children we're starting to see other signs and symptoms that are actually vascular issues. We see a much higher percentage of individuals having heart attacks, strokes, as a result of the coronavirus infection, okay? And this partially explains why it is that people with late stage infection with the coronavirus are only marginally helped by ventilation, by the ventilators, right? After a while, the problem is not the lungs, the problem is the vascular system, okay? So this is starting to explain why certain kinds of stabilizers, when it comes to heart attacks and things like that, ACE inhibitors and statins are actually protective for people that have contracted the coronavirus to some extent. So we can talk more about it. If you have questions, be happy to talk about it with you. But in this case, a tissue tropism might be, okay, this virus infects just respiratory tissue. This other virus infects the liver, right? Like with the hepatitis, for example. So you have specific tissues within the body that are affected. So now what we can do is we can turn our attention to animal hosts, right? I want you to notice the similarities here. So here we have a flu virus. All right, so they switched it to a virus that infects animals. And here we have a eukaryotic cell. I know that because it's got a nucleus in there, right? But notice that the steps are very similar, okay? We have attachment, just like we saw with viral proteins attaching to the bacterium. We have penetration, just like we saw with bacteriophage penetrating their genetic material into the cell. Now here they have this extra step, this uncoding step. But then we have the biosynthesis that we talked about before, where proteins and genetic material are replicated and produced. We have assembly. Well, this is just a little twist on maturation, right? Maturation was really just assembly of viral particles. And then we have release. Well, that's basically the lysis step, right? So yeah, they've changed a couple steps, but basically it's still all the same stuff going on, whether it's a virus for an animal cell host or a prokaryotic cell, okay? Attach, penetrate, biosynthesis, maturation, which involves assembly, and then the lysis, which sometimes they'll call the release of those viruses, okay? All right, and again, just like we would see in bacterial cells with prophages that incorporate their genetic material into the host cell, this can also happen with animal cells. Don't get too caught up with the details on this image. I'm not going to throw this diagram at you and freak out on the test or something. But what I will bring your attention to is that this is HIV. Okay, HIV is an enveloped virus. It is attaching specifically, and this cell absolutely has a tissue tropism. HIV attaches to helper T cells, really important cells in the immune system. Okay, it's not infecting, liver cells or kidney cells. It likes this one particular immune cell. Okay, so it has a tissue tropism. It's attaching. It's penetrating. Right, it's getting that genetic material in there. And this particular virus like some prophages. The phage means it's for bacteria, right? These viruses attack bacteria. But here we don't say prophage, we say provirus. So we use the word virus here when it's an mammalian cell and a virus affecting an animal eukaryotic cell. So this is very similar to a prophage with bacteria. So it incorporates itself, and this starts to explain why it is that someone can be infected with something like HIV. And then we have this gap in between the penetration step and the biosynthesis and getting the ball rolling to actually cause all of the signs and symptoms of this virus. Because in between, you can have this incorporation into the genetic material of helper T cells, and then helper T cells divide and perpetuate the genetic material of the virus. So what we say is sometimes that someone has a latent infection. There's a dormancy period where the virus doesn't actually cause signs and symptoms that we typically would associate with that virus. It's kind of hiding away. It's like, it's sometimes when we use these terms, latent infections, they're not always caused by a provirus like this. Some latent infections, like let's say chickenpox, it doesn't actually incorporate itself into the DNA of cells. It hides out along our spinal cord in our nervous tissue. Okay. Let's see. There are others, but so I'm just what I mean to say here is that latent infections, one way people could have a latent infection is because of proviruses like this. Okay. Versus chronic infections, this would be a situation where the virus is causing the signs and symptoms from the get-go and for a long period of time. There's no gap. Okay? And I thought we'd just stop with a little image here of the HIV virus. You can see here that this HIV virus is an enveloped virus here because there's that membrane around it. Okay? All right, so I think that's about it, guys. This would wrap up this whole section for Module 1. And be sure to tune in and send me an email. We can set up a one-on-one conversation on things if you want. Okay, so I will be in touch with you guys.